Hi everyone, today we're going to continue our Campbell Biology review series, and today we will be discussing vascular plant structure, growth, and development. We will split the videos by concept for each chapter to make them easier to watch, and all figures used are from Campbell Biology unless otherwise noted. So first of all, what's a vascular plant? They're plants that are characterized by specialized vascular tissues such as xylem and phloem for the transport of water, minerals, and food. And usually they can be split into two systems, like in this basic plant. They can be split into the root system and the shoot system. So let's talk about the root systems. Essentially, roots have there's a big primary root, or not big, but we'll get to that later. But in this case, there's a main primary root you can see that is referred to as a taproot here. There's going to be another type of root called the fibrous root system, but we will get to that later. So the taproot is this big main root, and then there's a bunch of lateral roots that function in water absorption. So while the taproot is for structure and anchoring, the lateral roots are going to absorb water. And what's important about this is that many people think that roots are mostly for anchoring, but the main job that they have is to absorb water and minerals. So let's go to the two different types of systems, the taproot system and the fibrous root system. You can see here in this carrot that the taproot is, well, the carrot, and it's really big relative to the lateral roots you can see here on the sides. And so a taproot, while energetically quite expensive, is beneficial to the plant and most dicots that it can provide stability and act as an anchor, as well as be modified to do a variety of other different tasks that we'll see later. And so that's for most dicots. In contrast, in most monocots and other smaller vascular plants like herbs, the fibrous root system is consistent of a mat of equal sized roots. And what's different here is the primary root. So this is the PR, the primary root. Here, there's no primary root. Why is that? Because the primary root dies off early in these plants. And so instead, small adventitious roots emerge from the stem. So you can see here there's a direct connection here. So adventitious means a plant organ arising from an unusual location. So in this case, the roots would be considered adventitious because the stem is an unusual location to arise from. So let's go back to some modified roots here. You can see here in this radish that the seedling is growing a taproot for a big anchor and for stability, and then there are little hairs coming off here. You see that? That's to increase the surface area to volume ratio. And in this corn plant known as Zea mays, the roots act as a suspensor root for the main big stalk that you can see here. So it's going to provide additional support. In this mangrove, you can see that the roots are growing above the mud. They are shaded in here. They're going above the mud, and that's going to help them absorb more O2 from the swamp surroundings. And then finally, in this beet, the taproot has been modified to store energy and food. Let's go to stems now. Stems, one part of the shoot system with the other part being leaves, have a node-internode structure. So a node is defined as where the points at which leaves are attached. You can see here. And then you can also see that the internode is where it goes from one intersection to another. And most photosynthesis is conducted in the leaves, but the stem may also do some of it. So in this structure, there are two main things I wanted to point out here. Before moving on to the leaves, you see how there's a bud here and a bud here? These are called apical and axillary buds. The difference is that apical, which stands for terminal bud, is the bud at the tip of a plant stem. So you can see here, it's also a tip because this is also a stem. On the axillary bud, however, It's a bud that can form a lateral shoot, which is this thing, and it appears at the angle between the stem and the leaf. So imagine if there's a pretend angle here, that's where the axillary bud is going to be. And then some stems are again modified to store food or to reproduce asexually, as you can see here. In strawberry plants, the stolons or little runner stems help with, the plant to reproduce asexually. And in potatoes, the tubers, commonly mistaken for roots, are there for storage and once again are modified stems. So now let's go back to leaves finally. And leaves, we can see here in this relatively basic model that these are what we call simple leaves. So there's another type of leaf called compound leaves, and we'll show them right here. For simple leaves, you can see that there's one big undivided blade, and there's a petiole marking the point of intersection, and then there's a bud here that marks that it's a single leaf, sorry. But here, on the compound leaf, there's several leaflets on one whole quote-unquote leaf. And then we can see here that this leaf structure, quote-unquote, is marked by this bud presence. And again, the petiole is the same function as before. So that's how you can distinguish them from the compound leaf itself. From each individual leaflet. So now let's go to tissue systems in a plant. There's three types of tissues in a plant. There's dermal, ground, and vascular. So let's go one at a time. Dermal tissue is a protective outer covering of plants. It normally consists in non-woody plants, so small herbs, grasses, things that aren't trees really. Those plants are going to have an epidermis. And they're going to have really a single layer of cells protecting them from the outside. In woody plants, such as trees, they're going to have a periderm, which replaces the epidermis during secondary growth. And secondary growth is when the tree becomes fatter, it becomes wider. The periderm is made of the cork cambium, and it's basically bark, but without the main phloem. So it's basically the outer covering of the tree. And then there are other parts, such as the trichome. You see these, so basically these little hairs, and this picture is from a Wikipedia article on trichomes. These little hairs are what we call trichomes, an epidermal cell that looks like a hair-like outgrowth on a plant shoot. It can help reduce water loss and reflect excess light. And finally, they can also help defend against insects. So in some plants, such as the sundew, the bug will get stuck on the sticky trichomes and be unable to move. And so that's how it helps defend against insects. Now let's go back to our vascular model. Let's go to ground tissue. Ground tissue is tissue that is neither vascular nor dermal. It can perform storage duties. So let's write that down. It can perform storage, photosynthesis, support, and short distance transport functions. And the other part of ground tissue would be the pith. This is the ground tissue internal to the vascular tissue, and the cortex would be external to the vascular tissue. So let's consider the ground to be pith inside vascular, and then cortex outside vascular. So now let's go look at the final one we have left, vascular tissue. Vascular tissue, it facilitates transport of materials to the plant to provide mechanical support. And they mainly take the form in most plants, the xylem and phloem. And the xylem is going to conduct water and dissolved minerals up and down the plant to where it's needed the most. While the phloem is going to transport sugars from sites of photosynthesis, so the leaves, down to the sugar sinks. So remember the potatoes we saw? Those are what we could consider a sugar sink. They're going to get most of the sugar and act as a storage. So it's going to go down the shoot and into these roots. So now we covered the types of tissues, but let's go to the types of plant cells as well. The types of plant cells are parenchyma, collenchyma, and sclerenchyma. So let's look at each one individually. Parenchyma cells perform metabolic functions. They're really thin, they're flexible, they have primary cell walls with no secondary cell wall. They have a large central vacuole, so all this blank space you see here, that's all the central vacuole. And they can have chloroplasts, which photosynthesize, and or amyloplasts, which store starch. They can divide and differentiate. Collenchyma cells, on the other hand, support young plants of the plant shoot, which is non-root part, remember? They're elongated cells with unevenly thickened primary walls. So that's why there are places where this side of the wall is thicker, while in other parts it's going to be relatively less. And they're flexible and elongate with the growing leaves and stems. So as the plant grows up, these cells will also start growing longer. And then finally, we move on to sclerenchyma cells. These are rigid supporting elements of the plant. They contain secondary cell walls and are produced after elongation. So after the plant grows in length, these cells are produced, and they contain a lot of lignin. And lignin is a strengthening polymer found in all vascular plants. And when they reach maturity, when sclerenchyma cells reach maturity, they cannot elongate. So essentially, this also means that they're dead at functional maturity. So after the secondary cell wall is made, they die. And they occur as fiber, and they give the gritty texture to pairs in scleroid cells. So finally, we arrive at the xylem and the phloem. Let's look at the xylem first. The xylem has tubular, elongated parts that are dead and lignified at functional maturity. So this is important, they're dead. And tracheids are in all vascular plants, so all VP, and they're long, thin, with tapered ends, with water moves from cell to cell through the pits. And vessel elements are in most angiosperms and some gymnosperms. And basically, they're going to be shorter, thinner, and less tapered than tracheids. And the end of each element has a perforation plate, seen here, that enables the passage of water. And when they are put together, they are arranged into long pipes called vessels. So now let's look at the phloem parts. These parts are alive at functional maturity. And in seedless vascular plants and gymnosperms, the phloem is going to be made out of narrow sieve cells, which are shown right here. And essentially, in angiosperms, the phloem is, however, going to be made of sieve tube elements, shown here. Though alive at maturity, sieve tube cells have no nucleus, no ribosomes, no vacuoles, or even a cytoskeleton. But however, they still remain alive. And sieve plates exist between the elements, shown here, to allow fluid flow. And another part of the phloem is going to be the companion cells. You can see them here and here. And they are non-conducting cells connected to sieve tube elements via plasmodesmata and function to help the elements live. And they can also assist in sugar transport. But again, the main parts are going to be the sieve tube element and the companion cells. That'll be all for this concept. Stay tuned. Thank you for watching.